Translation in prokaryotes. In prokaryotes, no nuclear membrane separates DNA from cytoplasm and its ribosome. Thus, the process of translation or polypeptide synthesis begins even before mRNA synthesis is completed. This process is called coupled transcription translation. It does not occur in eukaryotes because transcription occurs inside the nucleus and translation in cytoplasm. In prokaryotes, protein synthesis occurs in three stages: initiation, elongation and termination. Let us discuss the stages one by one. Now, we can discuss on initiation. Initiation of protein synthesis consists of two processes: formation of 30S initiation complex and formation of 70S initiation complex. First, formation of 30S initiation complex. It requires the following process: a strand of mRNA, the initiation factors like IF1, IF2 and IF3, and guanosine triphosphate, that is GTP, a 30S subunit. Since in the cell nearly all ribosomes exist as 70S particles, the dissociation of 70S particle requires the initiation factors IF1 and IF3. 70S ribosome, 50S plus 30S. This 30S binds with IF3 and plus 50S. The IF3 primarily acts as anti-association factor. IF1 increases the rate of forward reaction. Now, formyl methionine tRNA or fMet tRNA. A specific tRNA called fMet tRNA brings a modified methionine to 30S initiation complex. tRNA F has a different sequence than the tRNA that inserts methionine in internal position of the peptide chain. Both methionine and N-formyl methionine are linked to tRNA by same amino acid tRNA synthetases. Please see the picture. Steps in the formation of 30S initiation complex. The 30S subunit is associated with IF1, IF2 and IF3. IF3 is required for binding mRNA to 30S subunit. The 30S subunit binds on purine-rich Shine-Dalgarno sequences in mRNA, which is either identical or similar to the sequences AGG, AGG, U. Base pairs with the pyrimidine sequence in the 16S rRNA. The Shine-Dalgarno sequence lies within 10 nucleotides of the initiated codon. It places the initiated codon in the appropriate position in the 30S subunit to bind to the initiated tRNA, fMet tRNA. To combine tRNA to 30S subunit, IF2 and GTP is required. IF3 dissociate from 30S subunit upon the binding of fMet tRNA. Formation of 70S initiation complex. The release of IF3 allows the 50S subunit to bind to the 30S initiation complex to form the 70S initiation complex. The GTP is hydrolyzed to GDP and an inorganic phosphate upon the formation of 70S initiation complex. Then, the initiation factors, that is IF1, IF2 and the GDP and the inorganic phosphate are released from 70S initiation complex. There are three sites on 70S ribosomes: the amino site, that is A site, peptide site, P site and ejection site, that is E site. The P site is occupied by fMet tRNA during initiation. Elongation. In this phase, the amino acylated tRNA, that is complementary to the codon adjacent to initiation codon, that is AUG, is inserted in the A site, starting the process of elongation. Three elongation factors EFs, that is EFTU, EFTS and EFG are required for elongation. EFTU is helpful for the delivery of an amino acid tRNA to the empty A site on the ribosome. GTP bound to EFTU is hydrolyzed at this time. GDP from the hydrolysis of GTP remains associated with EFTU until displaced by EFTs. The EFTU-EFTS form a complex that is split by binding of another GTP. providing an EFTU GTP complex for the delivery of next amino acyl tRNA. EFTU GTP delivers all amino acyl tRNA except FMET tRNA. Please see the picture and table. The peptide bond formation. The activated amino acid attached to the tRNA in the P site, initially FMET tRNA, is transferred to the amino group of amino acyl tRNA in the A site. Formation of peptide bond is catalyzed by an enzyme called peptidyl transferase, which is a 16S rRNA, an integral part of 50S subunit. In this reaction, two amino acids are attached to the tRNA, dipeptidyl tRNA, in the A site and leaves an uncharged tRNA in the P site. Now, the translocation. The movement of ribosome in a 5' to 3' direction along the mRNA is called as translocation. This translocation is catalyzed by EFG. The EFG forms a complex with GTP, which is hydrolyzed to GDP and PI. This movement results in the following process: release of uncharged tRNA from the P site, movement of the dipeptidyl tRNA to the P site, and formation of unoccupied A site. Now, termination. Elongation continues until a termination codon encountered. A release factor is responsible for termination of translation. Stop codons are recognized by the release factors. RF1 recognizes UAA and UAG. RF2 recognizes UAA and UGA. The third release factor, that is RF3, in association with GTP, promotes termination. The binding of release factor induces peptidyl transferase to release the peptide from the tRNA from P site by hydrolysis. The tRNA is also released from P site. The ribosomal subunit then separate in a GTP hydrolysis dependent manner. The 30S subunit may move along the mRNA until another Shine-Dalgarno sequence is encountered and translation resumes, or it may completely dissociate from the mRNA.